Good afternoon. It is a true pleasure to welcome Dr. Elaine Fuchs to Vanderbilt as a participant in the Discovery Lecture Series and as the very first speaker of the Lloyd E. King Jr. Lectureship. Dr. Fuchs is the Rebecca C. Lancefield Professor of the Rockefeller University, is within the Howard Hughes Medical Institute in the Laboratory of Mammalian Cell Biology and Development. She is in the midst of an absolutely brilliant career that began at Princeton, grew at University of Chicago, and continues now at the Rockefeller. Among too many awards, honors, and presentations to mention, as after all we want to listen to her this afternoon, she is a member of the National Academy of Sciences and the Institute of Medicine. Her highly productive research interests are centered in cell and developmental biology, with an enormous interest in the skin as the model organ. We in dermatology owe her a tremendous debt of gratitude for her productivity and her insights into cutaneous biology. And it's very fitting, therefore, that this Discovery Lecture is coupled with a Lloyd E. King Lectureship to honor Lloyd's long-term dedication to building dermatology at Vanderbilt. He spent 25 years as the chief of dermatology and continues to contribute today. So with that, Elaine, we're pleased to welcome you to Vanderbilt and are eager to hear your insights on stem cells and their lineages in skin.
Well, I would just like to start by saying thank you. I really feel that this is a tremendous honor. Not only was I excited to come here, again, I think it's been about 5 years since I've been here last time. Last time invited by the students, and this time being the first lecture of the Lloyd King Lecture Series, I think is a particular honor for me because when I came from Princeton to MIT to start my postdoctoral career many years ago, the very first experiment or the very first assignment that I had when I entered Howard Green's laboratory was to take this group of rat submaxillary glands and purify epidermal growth factor, and I was shoved into my face the King and Cohen papers and asked to go and purify enough for the lab for the next 10 years. So that was everyone's postdoctoral assignment when they first arrived. But being trained as a biochemist, it was what endeared me to Howard Green because then thereafter I became the person to purify epidermal growth factors. So Lloyd, I can attest that your protocols and methods work very well. So I left the field of epidermal growth factor after I purified EGF from rat submaxillary glands, and I began to work on keratins. I thought what I do today is to tell you a little bit about the work that my laboratory has been doing since my move to Rockefeller University predominantly. And here we've begun to really focus our attention on what got me into becoming an epidermal biologist in the first place, and that is to focus on the stem cells of the skin. And so I thought I'd start by reminding all of you in the audience. Lloyd doesn't need a reminder, and George doesn't either, but to remind the rest of you that, in fact, you have been as interested as me, perhaps, in skin all of your life. So clearly, in terms of thinking about skin, nature clearly has had a lot more fun and fancy in creating body surfaces than she has in creating any of the ugly organs that are tugged underneath it and that most of you use as model systems for study. And in fact, nature has given us a certain set of interests in terms of skin biology, and that we as human beings throughout all cultures of the world have continued to adorn upon what nature has given us, often using the appendages of other animals for that adornment. So my laboratory works just a little bit deeper, in fact, not very much deeper into the skin, looking at the epidermis and its notable appendage, which is the hair follicle. And for those of you who need a reminder about what this structure at your skin surface is good for, if you didn't have an epidermis, you'd rapidly dehydrate and die. And if you survived that process, you'd die from infection. So the epidermis is only cellophane thickness in the body, and yet it provides an important barrier to keep harmful microbes out and essential bodily fluids in. In addition, the epidermal appendages provide warmth and protection and also lubricants. And all of these different epithelial tissues of the skin are derived from a common embryonic skin progenitor cell, a stem cell, if you will. And in the adult skin, every 4 weeks' time, you've got a brand new epidermis on your body's surface. And of course, if you're lucky, throughout the course of your lifetime, your hairs not only undergo periods of rest, but also periods of growth. And so what all of this dynamic changes in equilibrium mean is that in the adult, there have to be populations of stem cells that exist in the adult skin to allow this continual rejuvenation at our body surface. And so we use skin as a model system for studying stem cell biology because it is a readily accessible source of stem cells. Its lineages are spatially and temporally well defined. And then finally, adult skin stem cells can be cultured and they have clinical potential. And this is really what prompted me, trained as a biochemist, interested in interfacing human biology with medicine. This is really what interested me about the epidermis and continues to serve as a really terrific model system for studying. So like any good stem cell, the stem cells of skin have the capacity to divide to generate self. They exist as slow-cycling progenitor cells in the skin. They express keratins 5 and 14, which are the keratins that I cloned a number of years ago. They give rise to rapidly proliferating progenitor cells as well. And then at some point along the lineage, the point of no return may vary with regards to different stem cells. And in fact, this is an issue that is a question in all of stem cell biology. But at some point along this lineage, then these rapidly proliferating progenitor cells undergo differentiation to become either, in our case, the epidermis, the sebaceous glands, or the hair follicles. And really the overall problem that we're dealing with is one of morphogenesis, and that is how do you start with a stem cell and build a tissue? And then if you can understand that process, how do you start with a stem cell and have the capacity to build different types of tissues? And that's really the central problem that my laboratory focuses on. So several years ago, we felt that if we wanted to be able to examine the behavior of stem cells in the skin and to identify them, then we needed to have a way of labeling them and tracking them. And at the time, all we knew about the stem cells in the skin is that they're relatively slow-cycling cells, typical of stem cells. They're used sparingly and hence divide infrequently, and that they express keratins 5 and 14, which were characteristic of all the mitotically active cells of the skin. And so we made 2 transgenic mice, the first expressing a tetracycline regulatory transcription factor under the control of a keratin promoter, and then the next, a histone GFP under the control of a tetracycline regulatory element. So now, when these mice are mated, the gene is on, the mice turn green, and a cross-section of the skin shows us that all of the nuclei of the skin epithelial cells, but only the epithelium, have expressed histone H2B GFP, and that includes the putative stem cell compartment of the skin. And so all we have to do at this point then is to start to add tetracycline to the animal's diet, and now the gene turns off, and if we wait long enough, the rapidly dividing cells dilute out the label, the differentiating cells are lost from the skin, and what we're left with is just this infrequently cycling population of keratin 5, keratin 14 positive cells, And those cells reside in a region of the hair follicle known as the bulge. It's located just below the sebaceous glands. And you can really think of the skin as being subdivided into micro units, each composed of a hair follicle, surrounding epidermis, and associated sebaceous glands. And so this now allows us to monitor the behavior of these cells. So let's first look at what happens at the initiation of each new hair cycle. And what happens is that the epithelial cells become activated by mesenchymal cells or dermal papillae cells in the dermis, and these cells then stimulate a new growth of the new hair cycle. And you can see this new hair follicle starting to grow downward. What you can see is that as the interactions occur between the mesenchyme and the epithelium, that very few of the label-retaining cells that exist within this compartment are activated. As they leave the niche, they undergo rapid division and have diluted out the label. And we know that these cells down here are indeed derived from the bulge. They do have fluorescence. It's just much less fluorescence than the bulge cells. So this already is telling us something about what happens at the start of each hair cycle. Only a few of these stem cells become activated, and then they give rise to the bulk of the tissue. That's another common property of stem cells. If we now take a look at what happens in an injury response, we can scratch the animal's epidermis, and now you can see many more of the cells become mobilized and move upward. They move upward at the same time they're dividing, so they do dilute out the label, but many of them are moving upward now, and perhaps suggesting a greater sense of urgency or response of these stem cells in response to injury as opposed to normal homeostasis in the hair cycle. And so the next thing we can do is to take advantage of the ability to be able to use fluorescence-activated cell sorting to purify these cells, and this now allows us to place these cells into culture. And if we first take the label-retaining cells with very bright fluorescence and we put these cells into culture, they give rise to large colonies composed of relatively small undifferentiated cells. And if we then pick a single colony derived from a single label-retaining cell and passage it, we find that we get more large colonies indicative of self-renewal. If by contrast we now take cells that have less fluorescence, that is to say their progeny, put those cells into culture, they tend to give rise to smaller colonies composed of larger, more differentiated cells, and on passage, those colonies undergo senescence. So these data suggest that the label-retaining cells that we've identified do have the properties of stem cells. So now the question is this a reservoir of multipotent stem cells? Because you could argue a priori that we simply have a bag of stem cells, and some of those cells get mobilized to repair the epidermis on injury. Others, a different population of cells, go on to give rise to the new hair follicle. And so in order to be able to address whether one cell can do all of these different things, we have to use clonal analysis. But in this case, we have our experiment because we can now take the contents of a colony derived from a single label-retaining cell. These are GFP-labeled positive cells from the histone H2B GFP. We can then combine them with unlabeled dermal cells from a wild-type mouse and then graft them in a full-thickness graft onto a nude mouse that's defective in hair follicle formation. So if you just take the dermal cells alone, you actually learn something very important, and that is that the epidermis can repair itself even though you've undergone a full-thickness wound. So what these data tell us is that the epidermis from the nude mouse can repair the wounds. That tells you that there must be at least unipotent stem cells or progenitor cells that exist within the epidermis. And that's an important concept that is worth bearing in mind. It's also a good thing because when you think about evolution, you wouldn't have wanted nature to be able to have no backstop about being able to repair wounds in areas that don't have hair. And so there has to be some mechanism, and that mechanism appears to be the existence of unipotent stem cells. If, on the other hand, we now take the contents of our single label-retaining cell and combine those with the unlabeled dermal fibroblasts, you see these tufts of hair. The hairs are green. They might do pretty well in one of those country music bars here in Nashville. We could tailor-make this to whatever fluorescent color you'd like. It wasn't quite the intent of the initial research that we were doing here, but it did give rise to a number of different emails and phone calls from a number of people, not necessarily scientists, who suddenly took a tremendous interest in our research. So as scientists, we tended to focus on the biology, and here you can see green epidermis, green sebaceous glands, and green hair follicles. And since all of these hairs and all of these structures are derived from one single label-retaining cell, that addresses the question, these are multipotent stem cells that exist within the hair follicle. So I'd like also to just spend a few minutes talking about this issue of unipotent versus multipotent stem cells. In fact, some people would not even argue that there is such a thing as a unipotent stem cell, but effectively a progenitor cell that has the capacity to generate the tissue. So I've just given you evidence that there have to be cells within the basal layer of the epidermis. That's the mitotically active layer of the epidermis that gives rise to the epidermis. So in fact, we've known for quite a few years that in addition to the fact that nude mice can repair their wounds, that the epidermal basal layer, the innermost layer, is the compartment of proliferating cells within the epidermis. The work of Chris Potton and his co-workers in the 70s and 80s suggested the existence of an epidermal proliferative unit that existed within these basal cells that could give rise to other basal cells. Fiona Watt and her co-workers used cultured keratinocytes and identified a population of beta-integrin positive cells that had higher proliferative potential than the beta-integrin low-expressing cells. But to this date, we still have no marker biochemically that distinguishes one basal cell from another basal cell in vivo, and thus at the moment, that's a challenge in the future as to whether or not there really is a difference within the basal population of cells between one and another. Are there really specific stem cells that exist within the basal layer, or are all of these cells effectively progenitor cells? In the sebaceous gland, we have made progress in answering this question, and here we've identified a population of cells at the base of the gland, and this is work by Valerie Horsley, a postdoctoral fellow in my laboratory. And in a genetic screen, we identified a protein BLIMP1, first identified in immune cells, that has a that by immunofluorescence labels a very small number of cells right at the base of the sebaceous gland. We did genetic lineage tracing, whereby we took a BLIMP1 promoter-driving Cree and combined that with the ROSA26 Fuchs gene having expression of YFP, so that in the presence of these two genes, what happens is that the BLIMP1 positive cells are the cells that now become YFP positive, and if in fact there are progeny for those BLIMP1 positive cells, they all become YFP positive, and the entire sebaceous gland becomes yellow. So these data argue that these BLIMP1 positive cells at the base of the gland are indeed progenitor cells that can give rise to the entire gland. We've also used BLIMP1 positive GFP knock-in mice to be able to purify these cells from the gland, and if we put these cells into culture, we give rise to sebocyte colonies that have BLIMP1 positive cells, a mixed oil red O positive sebocytes. Again, reflective of the unipotent progenitor status of these cells. So now, if we knock out the BLIMP1, you would expect a priori that if these are really progenitor cells, then perhaps we're not going to get any glands at all. In fact, what we see are enlarged glands. So what's happening here? Well, here what we see is that there is an elevation of c-MYC in these sebaceous glands that are enlarged, and that's interesting because BLIMP1 in the immune system serves as a repressor for c-MYC. We went on to demonstrate that that's also true in the sebaceous gland, but now why do we see in the knockout an enlarged population of these c-Myc positive sebaceous glands? Well, the answer came from doing pulse chase experiments, and here what happens is if you ablate the BLIMP1 positive cells, now what we see is the multipotent stem cell reservoir, the bulge cells within the hair follicle, now become BRDU positive. They start to proliferate, and the region between the bulge and the sebaceous gland base now turn out to be labeled. And so these data suggest that there's a precursor-product relationship that as long as you don't ablate the BLIMP1 positive cells, the BLIMP1 positive cells give rise to the gland. But if those cells are now damaged in some way, as we've damaged them genetically, now the multipotent reservoir gets mobilized and goes in and refuels the gland. We can further demonstrate this precursor-product relation by virtue of isolating the bulge cells and doing skin grafts, and on those we can generate BLIMP1 positive sebaceous glands. So these data really give us a good view, and I think one of the few views that we have in the stem cell field of precursor product relationships between a multipotent reservoir that is typically used in the normal situation for homeostasis of the hair follicle versus what happens when unipotent reservoirs are compromised. So what I've told you thus far then is that the bulge is a residence of infrequently cycling, undifferentiated, multipotent stem cells, and that now the environment has to change in order to activate these stem cells. In a wounding stimulus, there's repair of the epidermis, and if we ablate the progenitor status of the BLIMP1 positive cells in the sebaceous gland, there's sebaceous gland mobilization of the stem cells to replenish the sebaceous gland. And then in the normal homeostasis, there's a mesenchymal contribution from the dermal papillae cells that activates these bulge cells to regrow the hair follicle. And so under normal circumstances, that is the function of these cells, but these cells can respond in injury when the unipotent stem cells of these other structures are compromised. So we've also used fluorescence-activated cell sorting to be able to purify these cells and subject them to microarray profiling. And here we've identified 150 upregulated genes that are preferentially expressed in these bulge stem cells relative to their progeny. Among them, prominently, are TGF-betas, studied very extensively here by Hal Moses, and also BMPs. And we've shown that both by phosphosmad2 and phosphosmad1, that both of these signaling pathways are active in the bulge. We've also done experiments with both TGF-beta1 and also BMP6 and shown that both of these growth factors are important in transiently withdrawing the bulge cells from their cell cycle when we're culturing these cells in vitro. So these data suggest that these molecules, these signaling pathways might be involved in quiescence. We also see that members of the Wnt pathway, specifically Wnt inhibitors, are upregulated in the bulge, whereas in the activated progeny, we see signs of Wnt positive acting members of that pathway. And that was interesting because we've been studying Wnt signaling in the skin for quite some time now, and what we had shown using a Wnt reporter mouse a number of years back is that at one stage of the hair cycle, and that is when the mesenchymal epithelial interactions are taking place, a few cells within the bulge turn blue. And so those data suggested that at most stages in the hair cycle, the stem cells are sitting there in a Wnt inhibitory or Wnt restricted status. And so we've gone on to demonstrate that, and I just point out or remind you of some experiments that we did quite a few years back now, back when I was still at the University of Chicago. And in 1998, we published this mouse. This mouse is actually less fat than this mouse, but it does have a lot of excess fur. It has many excess hair follicles, and what we learned from elevating the levels of Wnt signaling in the skin is that Wnt signaling is important in dictating the density of hair follicles in the skin. We also got from that mouse a number of phone calls from non-scientists, and I tried to explain that those mice do get tumors. Those tumors, we worked out the genetic basis for. They're called pilomatricomas. I'm an armchair dermatologist. Whenever we have a chance to match pathologies, we try to work out the genetic basis in humans. And here we showed that human pilomatricomas, which are virtually hairballs underneath the surface of the skin, hopefully that's close to being an accurate description. I'm not sure whether George might have a different description of them. But we went on to demonstrate that these are due to stabilizing mutations in beta-catenin. So too much beta-catenin and there's hair tumors. We had done gain of function and loss of function studies as well, looking at the stem cell compartment, showing that there is a role for Wnt signaling both in stem cell maintenance and also in stem cell activation. So that then led us to wonder what TCF3 is doing at other stages in the hair cycle when the Wnt inhibitors are coming into play. So TCF3 is the partner for beta-catenin in Wnt signaling, and yet it's present at all stages in the hair cycle where we only see signs of Wnt reporter gene activation at the stage at which the stem cells are activated. So could TCF3 have a function at other stages? And Huang Nguyen in my laboratory went back and showed that TCF3 is actually present in the early embryonic skin at a stage before any of the fates are specified. So these cells are effectively multipotent skin progenitor cells when the skin exists as a single layer. And TCF3 is downregulated as fates are specified, and then it's only found in the stem cell compartment in the adult skin. And so here Huang made another tetracycline regulatory mouse, this one that'll induce genes in response to tetracycline. She activated TCF3 and found that in the presence of TCF3, if you maintain TCF3 expression, you completely repress all three differentiation fates afforded to these cells. And you just get these masses of undifferentiated cells with a few islands of dead cells, effectively necrotic cells, in the center. And so these data argue that in the absence of Wnt signaling, TCF3 acts to maintain the stem cells in their undifferentiated state. So we also went on to do additional transgenic studies showing that the DNA binding domain and the Groucho interacting domain of TCF3 are required for these repressive effects, but that the beta-catenin interacting domain and the C-terminal interacting domain of the protein are dispensable. So you need the beta-catenin interacting domain to activate the stem cells, but this repressive effect that keeps these cells in an undifferentiated state is not requiring beta-catenin. So we also went on to genetically profile the target genes that TCF3 then is involved in repressing repressing the differentiated state of these cells. And we're still in the midst of some of those studies. We've been doing chromatin chip-on-chip studies in collaboration with Rick Young's lab in order to look at the chromatin-occupied genes as well as the beta-catenin occupied genes when the stem cells are activated. So what I've really told you with regards to beta-catenin signaling is that there appears to be a threshold level of beta-catenin TCF-LEF that is basically going to dictate the behavior of the stem cells. If there's little or no Wnt signaling, then the stem cells are maintained in a quiescent undifferentiated state. As we see elevated levels of beta-catenin, what we see is stem cell activation. In the experiment where we super elevated the levels of beta-catenin, we saw too many hair follicles, suggesting a role for beta-catenin in hair follicle fate specification. I didn't mention, but I'll do so now, what got us into Wnt signaling in the first place is their role in hair differentiation. And then finally, I showed you that at elevated levels, you eventually get pilomatricomas. We've also shown that the groups of genes that are activated along this pathway are different. In the state of stem cell activation, we've identified proliferation in transcription factor genes, and then latent differentiation. There's a different Wnt signal and different TCF left DNA binding proteins that activate a different constellation of genes. So we also began to get interested in this issue of where does this adult stem cell niche come from? Virtually many different tissues of the body have stem cell niches in the adult. Where do they come from, and how are they set aside? And so in order to be able to look at this question, we again focused on the embryo, looking at that single layer of epithelium and how this single layer of epithelium receives different cues, in this case adjacent Wnt signals from adjacent ectoderm and underlying BMP inhibitory signals from the incoming mesenchyme to collectively activate Wnt signaling and allow this early hair bud structure to form. And so in this study, what we showed is that the hair bud structure is in fact active for Wnt reporter gene activation. So Wnt reporter activity does occur in this very early stage, as well as at the stem cell activation stage. We can manipulate Wnt signaling in these keratinocytes in culture, where we can get a handle at the molecular aspects of this. And we've also identified some of the Wnt target genes. One of them is a negatively regulated gene, E-cadherin, that is downregulated in the early stages of bud formation. We can transgenically activate elevated levels of E-cadherin gene expression. You completely bypass the ability to make hair follicles. So we've identified, at least in this case, one gene that is absolutely essential as a Wnt target gene to be able to make this bud structure. And then we've also conducted fluorescence-activated cell sorting and microarray analysis to identify that not only is E-cadherin downregulated, as we had predicted from our target gene expression studies, a number of other genes are either down or upregulated that we already knew about. Beta-catenin, as Walter Berkmeyer's work would suggest, is actually upregulated at the transcriptional level. And then there's a whole group of other genes, amongst them being transcription factors such as LHX2, a homeobox gene. We had no clue that LHX2 was involved in hair follicle morphogenesis. An antibody shows you that it's really expressed in these early stages. And now what's interesting is we've identified a whole constellation of genes, and we can trace their developmental pathway. And as I'm showing you here by LHX2, here is the bulge, and LHX2 in pink is also expressed in the bulge. And so we can trace from the very early stages at which you say, I'm going to make a hair follicle, you can now trace how that stem cell compartment arises. And its first indication of existence occurs at this stage. And remember what I said is that this particular stem cell compartment of the skin normally just makes the new hair follicle, but it can be mobilized to make epidermis and sebaceous glands only in injury. We've also identified a number of extracellular matrix, actin remodeling, and cytoskeleton proteins that are also affected in this early stage of bud formation. And again, with our ultimate interest of understanding how do you go from this single layer of cells to forming a bud. And you might be interested in lung development or mammary gland development or a whole variety of tracheal morphogenesis. There are a whole variety of different structures in the body where one undergoes these changes in morphology that are the early vestiges of tissue formation. And we're hoping to be able to, by understanding this very simple process, be able to contribute to that field. And we can, of course, as we've done with LHX2, use gain of loss of function studies in mice to be able to demonstrate the importance of these proteins. So just as we're interested in how do you make an early hair bud, we're also interested in how do you make the epidermis. And again, we focus on the very early stages of going from the single layer to making a stratified epithelium. how do you maintain a single layer of dividing cells and multiple layers of differentiating cells, and how is this process set up? And so here, Terry Lechler in my laboratory, or used to be in my laboratory, made a GFP-centrin mouse that allows us to look at spindle orientation. And what we showed is that at this early stage, as you might imagine, all the spindle poles are aligned parallel to the underlying basement membrane so that all the divisions are uniform and equal and effectively symmetric divisions relative to the basement membrane. But as you start to first see the signs of stratification, now you see a perpendicular orientation of these mitotic cells, and this gives rise then to one superbasal cell and one basal cell. And here, using a GFP ensconcin mouse, ensconcin is a microtubule binding protein where we can watch or image microtubule dynamics in vivo in the animal, and here you can see one of these asymmetric divisions in the basal layer. So we've mapped out these differences between symmetrical and asymmetric divisions in the skin, and you start with the majority of them being symmetric, and by the end of development, about 75% of the divisions are asymmetric. And we've seen that this is not just a property of the epidermis, it seems to be a general property of stratified epithelial tissues. So we've also looked at how does this happen at a molecular level, and here we've taken a page from the fly neuroblasts, where in fly neuroblast biology, genetically, there's a group of genes that are important in asymmetrically partitioning the protein contents of the two daughter cells so that one daughter cell adopts one fate and the other daughter cell adopts another. We've shown, after cloning the mouse version of Inscutable, that all of these different proteins of the fly neuroblast are also expressed in the mouse epidermis, and they're there at the right place and time. PAR3 and APKC are apical relative to the underlying basement membrane. LGN and Inscutable form a crescent in only those cells that have entered mitosis and are going to undergo asymmetric divisions. We've identified a coiled microtubule binding protein called NUMA that also associates with these crescents and these asymmetric divisions. And in the last year, there have been 3 different groups, fly groups, identifying the fly equivalent of NUMA called MUD, and showing that MUD is genetically involved in the fly neuroblast. So there appears to be a number of different parallels. There also are some differences between the asymmetric division process of the neuroblast and that of the epidermis. What we were interested in is a whole variety of different knockout mice that we had generated over the years. Apical in the epidermis is relative to the underlying basement membrane, and that suggested to us that integrins might be playing a role in this process. And so we looked at our beta-4 knockout mice and our beta-1 knockout mice. And beta-4 is at the core of the hemidesmosomes that link to intermediate filament proteins. Beta-1 is in focal adhesions that link to the actin and microtubule cytoskeleton. And what we showed is that the beta-4 appears to be dispensable for the process of asymmetric divisions, but beta-1 is not. And when you mutate beta-1 integrin, now the crescent does not form in the right orientation. And so what happens when you lose the orientation of this asymmetrical crescent is that you also lose the orientation of the spindle, and now you start to end up with aberrant tissue structure as a consequence because the spindles are not oriented properly within the developing tissue. We went on to demonstrate that APKC and PAR3 really appear to be important in the early stages of the polarity, that we also did the same thing looking at cell-cell junctions. Desmoplakin, which links to the intermediate filaments, is dispensable. Alpha-catenin, which links to adherence junctions through the actin cytoskeleton and possibly the microtubule cytoskeleton, is not. And so it really looks as if these cell-cell junctions and cell substratum junctions that are important in interfacing the actin and microtubule cytoskeleton set up the polarity relative to this underlying basement membrane rich in extracellular matrix. That sets the scene then in order to be able to properly position the spindle And it's not unless the spindle is properly oriented that you can ever get a proper functioning tissue. And I think the overall mechanisms that we're dealing with, I suspect are important in understanding tissue morphogenesis in a variety of different tissues. It really sets up the scene nicely for how you'd basically partition all the mitotically active machinery in one daughter cell, namely the cell maintaining its contact to the underlying basement membrane, because in the basal surface of the epithelium, as well as the lateral surface, are the tyrosine kinase receptors. This actually, I think, dates back to some of Lloyd's early work, and then the integrins as well are positioned in this area. So now that we have antibodies to epidermal growth factor receptor, you can see that there is lateral and basal preferred organization of EGF receptor. We've also been able to recapitulate these asymmetric divisions by plating epidermal cells on a fibroblast feeder layer. And here you can see one of the members of the asymmetric apical crescent, and here you see the EGF receptor. And it really looks as if there are going to be these two cortical domains that in these mitotically active cells asymmetrically partition, certainly the proteins that are associated with the cortical domains. We don't know how many different proteins are associated, giving rise to two distinctly different, at the protein level, distinctly different cells at the end of the division. We've also been interested in Notch signaling because Notch signaling plays an important role in neuroblast differentiation. And in the neuroblast, it's the cell that gets the crescent that basically is the cell that would get activated Notch signaling. That in the epidermis would be the first suprabasal layer, and interestingly, that is where we see activated nuclear HES1, which is the downstream target of activated Notch signaling. So whether or not this process of Notch signaling is linked to asymmetric divisions, we don't yet know, but we have done gain of function and loss of function studies with Notch, specifically with the RBPJ, the DNA binding protein of Notch, and with superactivated NICT. And what we've shown on the basis of that is that this transition between the dividing cells and the suprabasal differentiating cells is in fact Notch dependent. So it's functionally important. Whether or not it's linked to this mechanism, we don't yet know. So we're really looking at the whole issue of homeostasis within a tissue in the case of the epidermis as being this balance between symmetrical divisions and asymmetric divisions. And you can then ask the question of what might control this balance. You can imagine if you have too many symmetrical divisions versus asymmetric divisions or vice versa, what might happen to the overall tissue organization. And this is going to be a very interesting avenue for us to focus on in the first place in the future. We're also interested in how the process of epidermal homeostasis might change during wound repair. So in wounding, you can consider that as being a transient case at which you've got to respond. The tissue organization has to respond, has to change, redirect itself toward the wound, fill in the wound, and then go back to the normal tissue structure. So how does that happen? And we've just begun to start to get a few vignettes into thinking that these processes are linked to asymmetric divisions. And along those lines, several years back, we identified a protein called ACF7, which we showed has microtubule binding domains and actin binding domains. And it localizes to the plus ends of microtubules, but only those microtubules that are stabilized at a wound front by focal adhesions. And we're going to need the lights really down. I wonder, can somebody operate the lights back there to turn them down? Do we know? Do I have a... Okay, great. So I want to just show you what happens then. So what you're going to see in the wild-type cells, so these are bundles of actin fibers converging on what would be a focal adhesion site. We're putting in EB1, which is a plus-end microtubule tracking protein. So you're going to be looking by virtue of, you'll be looking at microtubule growth. And in the wild type, what you can see is that microtubules really grow, they really track along the actin fibers. And in the absence of ACF7, their tracking becomes completely randomized. So what happens when you don't have microtubules paying attention to what actin is doing? Well, the other thing I'll tell you is that at a wound front, up comes APKC and PAR3, same players that are involved in apical organization in the asymmetric divisions. In the absence of ACF7, APKC and PAR3 are no longer redirected at a wound front. Stabilized microtubules, by judging by glutubulin, also localized to a wound front. In the absence of ACF7, they're not there. Golgi microtubule organizing centers, a whole variety of different things, redirect in response to a wound. And in the absence of this cytoskeletal cross-linking protein, that doesn't happen. And now when you look at cells that migrate in at a wound front, in the absence of ACF7, they just do their own thing. They traject and curl around, they go backwards, they go forwards, and you can see that they just don't stop when they fill in a wound site. And so you might imagine that there's going to be a very interesting phenotype, and we're currently in the process of doing the conditional knockouts that allow us to look at that. But we're really viewing this now as the notion that perhaps what's happening is that this set of proteins that's involved in asymmetric divisions might also be redirected toward a wound front in a transient situation when you're repairing an injury. And that then raises the question of what happens when you mutate these genes. And we have mutated alpha-catenin, which is one of those central players. And in the absence of alpha-catenin, there are multiple layers of dividing cells, sustained activation of the RAS-MAP kinase pathway. There's a progression to invasive cancer, squamous cell carcinoma that occurs. And now if you look at their movement, the movement of keratinocytes out of a skin explant in the absence of alpha-catenin looks very similar to what I just described for you in the loss of ACF7. It's randomized. These cells grow randomly out of a wound site. They almost look like cancer cells. And in response to the wild-type situation, where what you see, I need to stop this guy, what you see is a real directionality to the actin cytoskeleton in a normal wound front. And so we're hoping that we're starting to get a link between understanding normal tissue morphogenesis, transient repair and wound response, and then what goes wrong when this machinery becomes mutated. And we think there's a really strong link, and there is going to be a strong link to all of these proteins, or at least many of these proteins, in understanding transitions to cancer. So I think, without going into any more details, I'll just say that with regards to human squamous cell carcinomas, we've now looked at the parallels, and in fact, most of the squamous cell carcinomas in humans turn out to have downregulation or reduction in alpha-catenin. And of course, in a tumor where you've got completely disorganized cells within that tumor structure, you also have aberrant spindle orientation and many of the same things that I've just described to you that occur in the absence of alpha-catenin. And remember, alpha-catenin is central for positioning APKC and PAR3. So this has taken me quite a few years to get to the problems that we're interested in now. I started in Howard Green's lab at MIT back in the late 70s when Howard was working out the system of using cultured epidermal cells for burn grafts. And interestingly, this is effectively what we think is unipotent epidermal cells. They can repair the epidermis very nicely, but the skin of these patients does not sweat and it does not produce hair follicles. It raises the question of whether or not the multipotent stem cells within the skin might actually be beneficial in terms of improving the quality of use of cultured cells for burn operations. And that's something that we're currently working with a few plastic surgeons at Stanford. We've also been interested in the overall question of whether or not these stem cells might be able to do something besides simply make sebaceous glands, sweat glands, hair follicles. And along these lines, what we've done is to subject our skin stem cells to a process known as nuclear transfer, where you take an unfertilized oocyte, in this case of the mouse, remove the nucleus and discard it, and then replace it with our purified bulge skin stem cells, and now take these hybrid cells and culture them to the blastocyst stage, and here we've implanted them into a host mother. And what I'll show you then is that these mice are basically cloned mice from our skin stem cells. And we've now made a number of different mice, shown that you can reproducibly clone mice from skin stem cells. Those mice age, they give rise to normal litters, they're viable, and the litters now, we've kept the mice going, the oldest ones are now several years old, and they don't show any of the sort of dolly signs that the single cloned sheep has shown. And it could be that sheep live a lot longer than mice, but the skin stem cells that we've subjected to in these studies turn out to be better than any of the other skin stem cells that any of the other skin cells that we've used. And so we think that there might be an advantage to using the stem cells. So the work that I told you about today, Duina Tumbar and Geraldine Gouache did the work on the H2B-GFP mice that really cracked open the whole field for us in really tackling the issues of stem cells in the skin. Duina is now on the faculty at Cornell at Ithaca. Geraldine is still in my laboratory finishing off her studies this year. Terry Lechler did the work on the asymmetric divisions, and Terry is now setting up his laboratory at Duke University. Horace Ree did the work on LHX2 and tracing the origins of adult stem cells. Horace has finished his Ph.D. and is now finishing his M.D. Cedric Blanpain is an M.D. Ph.D. Who did the postdoctoral work with Bill Lowry on the multipotency of the stem cells in the hair follicle. Cedric has returned to the University of Brussels, where he's an assistant professor, and Bill is now an assistant professor at UCLA, taking advantage of UCLA's stem cell funds. Huang Nguyen is currently on the job market, and Huang did the work on TCF3 that I told you about. So with that in mind, I'd like to thank you all again for your patience. Thank you very much for the invitation and honor, and thank Lloyd in particular for his inspiration over the years with regards to my work in dermatology. So thanks again. Yes, Eric.
Could you comment on the essentialness of asymmetric cell division and the stem cell property, pluripotent and multipotent? It seems that if you study flies, it's extremely clear to some people, and you get the mice or other mammals, there are clearly, but there are stem cells that don't look like they divide asymmetrically. What is your take about it?
Yeah, so I think a priori there's no reason why you have to have asymmetric partitioning of proteins in order to get asymmetric fates of two daughter cells. You can envision a situation where you could have a division that is perfectly symmetrical with regards to partitioning of proteins, but if one of the two daughter cells finds itself in a different microenvironment, mini microenvironment, then that daughter cell could end up adopting a different fate. And so my own view is, although most people in the stem cell field believe that there's this existence of asymmetric divisions through asymmetric partitioning, my own view is that there's no reason why we should necessarily accept that. And there's very little evidence to cause me to want to accept that. Even within the work that I've described with regards to the skin, we're currently in the process of collaborating with Jürgen Noblecht, who's one of the fly neuroblast biology asymmetric division people. And Jürgen's lab and my lab, independently, unbeknownst to each other, had both cloned inscutable from the mouse. And as soon as we found out that we had both cloned it, we're getting together to collaborate to do the conditional knockout. And so Jürgen will do that in neuroblasts and mammals, and we'll do that in the epidermis. And until we've genetically demonstrated that it is essential or tested whether it is essential, we can't even make that conclusion. So at the moment, what we don't know is whether or not the notion of symmetric divisions followed by delamination is a backstop. It could be that that you've got these two separate mechanisms, and that effectively they're redundant mechanisms in order to get differentiation. My own view is that they're going to have subtle importances with regards to the thickness of the epidermis, wound response states, all of these other kinds of things. But at the moment, that's just speculation. So we're currently doing the genetic test, which will allow us to tell that. And I think once those conditional mice are available, that they should answer the question in a lot of other systems as well. But a priori, I completely agree with you that I don't think there's any reason to believe that that system has to be invoked. Yeah?
There are several million hair follicles. They are the gall cells of each hair follicle maintained from some external source, or they in an adult, freestanding and self-sufficient.
They appear to be freestanding and self-sufficient. So every single hair follicle has one of these compartments. What we've shown is that within those label-retaining cells, there are stem cells that are multipotent. What we cannot say is whether every label-retaining cell is in fact a stem cell. It could be that a number of those cells are simply, one, they may not be necessary, and secondly, they may not necessarily even be stem cells. So I think it's until one does transplantation studies directly from the fluorescence-activated cell sorting system, or until one has a way of directly isolating a single stem cell from the bulge, transplanting it and showing that with high efficiency that occurs, then we're not going to know the answer to that. And one of the problems is that because fluorescence-activated cell sorting, even in the best packed sorters, fluorescence-activated cell sorting does damage to the cells, it becomes very difficult to demonstrate that every cell within the bulge is actually a stem cell. But every bulge within the hair follicle appears to have stem cells, and those stem cells appear to have the properties of being label-retaining and keratin 5, keratin 14 positive. No, in fact, there does not appear to be any cells that are coming from the bone marrow. In fact, one can take the purified keratinocytes directly from the labeling methods that I've shown, and those cells appear to be the sole source of the tissue generation in the skin for the epithelium. And we don't think that there are, well, put it this way, there's no evidence to date that would suggest that marrow cells are providing in any way the epidermis or its appendages. Yes?
I
Yeah, so Musashi has been a gene that has been associated with stem cells. We do see an upregulation in Musashi in the stem cell compartment in the hair follicle, and we have not done any studies at all in my laboratory on Musashi, but there are certainly a number of labs that are interested in the relationship between Musashi and stem cells. So it all suggests that our stem cells might be similar in that regard to other stem cell populations. There's also a number of other similarities. CD34, for instance, is a marker of hematopoietic stem cells, and that's also up in the hair follicle bulge structure. But I would say if I look at overall where are the commonalities most clear, we would find them in terms of a role for notch and a role for notch and a role for Wnt signaling in the overall process of stem cell maintenance and stem cell self-renewal. And we would find it in terms of the TCFs and their role in maintaining a progenitor status. Those appear to be the major studies from our laboratory that would suggest commonalities between other systems. Yes?
I'm very interested in the stem cell labeling method, which can identify stem cells because they divide slower than progenitor cells. So, now people think cancer stem cells might be, there's a group of cancer stem cells that proliferate slower than other stem cells, which can survive like chemotherapy or radiotherapy and cause cancer recurrence. Do you think this method can use to identify cancer stem cells?
Well, put it this way, we're testing whether this method can be used to identify cancer stem cells. And I'd like to think that if there are cancer stem cells that have the properties of label retaining, then we will identify them in our various different cancers. So we've generated over the years a number of different models giving rise to different types of cancers in the skin. And we're in the process of doing the matings to be able to, and label retaining, to be able to test that hypothesis and be able to isolate those cells if in fact those cells do have that property. But it's really, it's an exciting question, and we just are in the early stages of those studies. We have, by the way, used this method and are further along on studies that we've done to look at other label retaining populations in different epithelial tissues. With regards to breast mammary stem cells, there were 2 reports in Nature in the last year that identified a population of label retaining cells that are K5, K14 positive that appear to be the stem cells in the mammary gland. And we are looking at other glandular epithelial tissues and are working on the parallels between those stem cells. Also looking at some other stratified squamous epithelial stem cells. There appears to be populations of label retaining cells in many different tissues.
Thank you very much.
Thanks.